So this is the power of DNA sequencing. And since your book was published, which at this point it's based on the perspective that was present about 10 years ago, since I'm using a book that's older, so therefore it's much less expensive to purchase. Since then, the importance of DNA sequencing as a component of microbiology, as a component of molecular microbiology, has just exploded. So when you go through this chapter, you'll see it talking about a lot of different types of ways of characterizing microorganisms molecularly. But although DNA sequencing is not missing from the chapter, both the huge emphasis that today we see on DNA sequencing is missing, and also the specifics of the modern ways that people go about sequencing DNA is missing. Instead, what we see are the old-fashioned ways of going about DNA sequencing in there, using gels and what have you. No one uses DNA sequencing gels anymore. I have some some glass that's used to support DNA sequencing gels in my laboratory that, in fact, I'm in the process of getting rid of, although, heck, they might be collector's items at this point. People would run DNA on these gels, and the longer the gel, the greater resolution you could get of the sequences that you were determining. And the more DNA sequence you could get per gel. And so this was really pushing the technology some 20, 30 years ago. And as a consequence, sequencing gels got longer and longer and longer and longer, and therefore the glass that supports them got longer and longer. I think the ones that are in my lab, the glass is something like, well, I don't know if it even fills up the screen, something like that long. I mean, they're big pieces of glass. But that's not the way people sequence anymore. In fact, the way people sequence nowadays is they take their DNA. Now, the DNA needs to be cleaned up before it's sequenced. And you have all sorts of what are known as kits to go about removing impurities from this DNA. But then you just send this DNA off to these sequencing centers, which have these machines, various types of machines, that are optimized to do various types of things, different read lengths of the DNA, etc. And what they do is they put the DNA into the machines, and then the machines sequence that DNA en masse, and then what gets returned to you is the actual sequence of DNA that has been determined by these machines. It used to be that we would sequence individual genes that way, and we would have to supply primers for that sequencing. We would have these precious primers in our precious DNA. They'd be sent off, and then we'd get back the sequence of an entire gene. These days, you send off entire genomes, and in fact, you send off multiple genomes that get sequenced simultaneously. And then you have to actually assemble these genomes, and we have computer programs that assemble the bits and pieces of sequence together into one larger genome. And then we've got programs that get you started in annotating the genomes, looking for where the genes are and where the control sequences of those genes are, etc. So, like I said, nowadays there is the study of microorganisms in a wet lab and a much more biochemical, with a much more biochemical perspective. And then there is studies that lead to DNA sequencing. And often the wet lab portion of DNA sequencing is relatively slight, because all you need to do is obtain that DNA in some manner and then clean it up and then send it off to somebody else, and they do the actual hard work of sequencing the DNA.